So thank you for assembling for Ethan's PhD defense. I'm going to do a quick intro. There's a lot of content to cover through, so I don't want to take too much time. This is always hard. I know many of you have done this before, of kind of a bittersweet sets of moments of defense, but also just celebrating years and years of work. Let me, to kind of share the philosophical thing, let me start with a thought experiment. All of you came from your offices, you opened up your laptops, you probably typed some emails, you probably browsed something. Since morning, 8 a.m. till now, you probably used 30 or 40 different software tools that have been built by open source community. If you think about every single day, what we use in our phones and what makes many of the internet services work, there has been this beautiful philosophy. And, I'm not saying many of the products that have been built on that philosophy then really scale it, but what you don't think about often are the individuals behind those who actually are the backbone of, and even till today, if you think that all of these large-scale companies would have patched up, that's actually not true. There is an incredible underground community of people that put blood and sweat to keep the ethos and the philosophy of in the open source community alive. You will meet one of those people today in this context of open science. And I think a question that we've been asking in the lab for the longest time is, why cannot that work for hardware? Why can't that work for instruments? Why can't that work for much of how we do science to begin with? And there are lots of challenges along the way, and I think this thesis really embodies that. But I think for the first few minutes, I want to focus on the person, and most of those people are thoughtful people out plugging away, thinking about these questions, and I think Ethan really embodies that personality. I think it's too complicated. Julios, there are some pretty pictures here that I will send to you later. And I think kind of they have been a remarkable anchor to an incredible amount of projects that we've done in this open space and science. And you'll see that. I see Benedict, one of our collaborators, is connecting on Zoom. I'll let you hit accept on that. And, you will, if you've known Ethan for the longest time, you would know and understand what I mean by this. Many a times these journeys take really a long time. It's not so clear that you can actually build a community around projects. A lot of times, the mountain that you're trying to climb to truly make tools work for others is very different from just making it work once in a lab. And I think many of you have seen that process and also seen that process realizing that that transition is very hard, and Ethan has been incredibly brave. They have taken that challenge multiple times, and this thesis is going to be filled with those sets of stories. I'll share some of the lighter moments of Ethan and their personality and how that led to many of those sets of projects. I have a tendency in the lab, and most of my lab members know this, to ship people on a very short notice to some part in the world that I haven't told them. That happened with Hadil, which 5 days ago flew to Kenya, and she's in Kenya. I don't know if she's connected. She didn't know she was going to Kenya, but it was important. We understood the point. And so I've done that multiple times with Ethan. This is a pretty out of place picture. And if I was to just show you this picture, you'd be like, what the hell is going on? There, Ethan is practicing one of these sets of shotguns that are a necessary requirement in the Arctic, because Ethan and Grace and several other people in the lab were in the Arctic for 45 days, a long time. And you get training to shoot not directly at polar bears, but scare polar bears. And, I think this was it's a project that we had been building and thinking about. Ethan and several other people stepped in, and it was quite transformative to a lot of work that happened afterwards. This is another picture now, Ethan in Rwanda. I had sent Ethan at that time for our collaborators in Tororo in Uganda for a lot of work that you'll also see on malaria. And then suddenly another group reached out, is like, oh, could you please come to Rwanda? And then Ethan was on a trip spending time in Rwanda. I don't have pictures from this, has also happened in building communities in Indonesia for another project that you'll hear a little bit that has now grown. And, I mean, I think to a certain extent, doing science out in the field is hard, but one of the key lessons that we learn, many of the tools that we talk about, you have to deploy them, learn where they fail. So you'll hear about those sets of failures and then what led to the sets of insights. I'm assuming you can figure out what these two people are doing. This is not searching for drinkable water. These are ice cores being made in the Arctic. And I think one of the threads that I like that Ethan has done in some sense is some really unsurmountable challenges that I have thrown. Ethan has been an individual who's actually tackled and faced those challenges, and you'll hear about that. I'll end with this story. This was one challenge that was not of my making. They created this challenge for themselves. This is Pranav and Ethan. We had decided we're going to take some drones out into the Arctic to do some deep sea underwater imaging. That's a story that Ethan is a part of that is not part of this thesis per se, but I'll just tell you the glitch. I have no responsibility to take in this. Ethan and Pranav are flying this glider. Now you have to understand this picture. That's the Arctic ice shelf. It's quite shallow because at the end of the day, you can only bring the boat out to a certain depth because these are icebreakers. So this is not very deep ice, so it's fractured. But to get a underwater drone for imaging, you need to be on the edge. And after some time, there is no orientation. So you have this thing moving around, you get an image out of it, everything looks the same. And both of them probably lost the orientation of what was going on. And at the end of the day, this drone actually got stuck in the propeller. Maybe at the end of the defense, we'll tell you what ended up happening, but the drone got at least the cables got chopped up. It's quite scary if something gets stuck in the propeller because you're out stuck there. And if the propeller doesn't work, it's very difficult conditions to go diving and fix the propeller, even if it's a small little cable. Fortunately for us, it was not an issue. The captain was really good about it, but we got an incredible data set out of this expedition, which is now going to be a story, something that nobody's actually seen in the Arctic. So I'm going to end here. Like sometimes sitting on the edge of the ice, ask yourself a question if you're going to dangle your drone, because some really remarkable things can happen, including you might jeopardize the ship that you're on. So with that as a note, let's get started. And I think thank you again for everybody for coming.
Going to try to get the Zoom screen sharing set up here. Okay, that is there. There, I hope. Oh no.
Ah.
Hold on. Okay. Yeah, screen sharing.
Looks good. Thank you.
All right, great. Yes. So thanks for the introduction, Manu, and thanks to all of you here for choosing to spend some of your precious time with me, whether in person, over Zoom, or via a recording of my talk, which is titled Building Capacity for Open Science Instrumentation, Global Health, Environmental Monitoring, and Beyond. So I'll open this talk by explaining the title line by line, starting with this technical term, open science instrumentation. Broadly, open science is a movement aspiring towards broader and more equitable societal involvement in the production and use of scientific knowledge so that science can serve all people and not just some people. UNESCO emphasizes 4 things as key elements of open science: open infrastructures, open engagement with society, open dialogue with indigenous and traditional knowledge systems, among others, and open access to scientific knowledge, including open access to scientific publications, data, educational resources, software, and hardware. So UNESCO specifically calls out software and hardware in this 4th element because producing scientific knowledge largely relies on tools for measuring and manipulating physical systems around us. So if we want to broaden engagement in those practices, then we'll need to provide broad access to the necessary software and hardware tools. So for example, over the past 2 decades, we've seen major increases in the accessibility of scientific computing in the life sciences, in large part through an open source ecosystem of Python libraries. Because these projects are open source, which means they're published in a way that allows anyone to independently use, study, replicate, adapt, and transform them, now a much broader population has access to computational tools for analyzing data, performing simulations, optimizing designs, among other tasks. This is just one example of the many ways that open source software has fundamentally reshaped the software industry so that people are learning from and building on each other's work much more easily and much more directly in the past and achieving new heights in what we can do. And we're starting to see parallel trends in the hardware world. So maybe the most recognizable example of this is with 3D printing, which until the late 2000s wasn't really accessible to ordinary people. What changed starting around 2005 was the emergence of an open source hardware project called RepRap, which built a community of people around the world to design 3D printers, which could be easy for people to replicate and easy for people to operate at home for manufacturing many of the objects needed in everyday life. In creating new 3D printer designs, people built upon lessons from prior efforts and also directly reused elements from previous designs, as we can see in this family tree of designs from 2006 to 2012. Many new design variants were created in order to adapt designs for different combinations of goals or constraints, such as portability or print size. The RepRap project's open source approach revealed and helped increase a demand for 3D printers for personal use. And after that, new companies such as Ultimaker and MakerBot then entered the market with affordable commercial designs, which directly built off of the RepRap designs. This made 3D printing much more accessible to ordinary people who wanted something more approachable for them as novices. So given such a strong impact of open source approaches on personal manufacturing technology, such as 3D printing, people are also excited to see if we can achieve similar transformations with scientific tools for measuring and manipulating physical systems, which I'll call instrumentation for this talk. People who develop instrumentation for open science often has goals such as improving scientific reproducibility and repeatability, encouraging equitable innovation, and facilitating research collaboration. The idea is to offer tools which other people can independently use and build upon in their local contexts, such as in this photo from 1973 of a health worker using a hand-powered centrifuge to process some biological samples to test for a disease in Guinea-Bissau. As just one of many more recent examples, here we see the open flexure microscope, whose structure is mostly based on 3D printed parts, and people have started to use it for imaging patient samples in pathology, as well as for monitoring soil biodiversity and health for agriculture, like we see on the right. But although projects like Open Flexure have demonstrated that open science instrumentation can work and can solve real problems, and although academic researchers are skilled at designing tools and publishing those designs, the impact of open science instrumentation on science is still very far from the impact that open source software has had on the software industry. For my PhD, I wanted to understand and address one aspect of the many challenges which have hindered our efforts so far, specifically in the problem of making new designs actually practical for many people to implement, adopt, and modify across the world. Previous researchers have already emphasized other important implementation challenges, like in the policy brief quoted here, and many of these are specific to the problems of manufacturing physical devices at a level that could compete with existing commercial instruments. My thesis takes a different direction by focusing on technical difficulties in the ways that we try to collaboratively create and modify designs. So unless we make open science instruments actually practical for many more people to modify in whatever ways they need, then their advantages over other tools will be much narrower beyond a small group of people, even if these other hardware-specific implementation challenges are solved. So we need to improve our collective capabilities for various kinds of instrumentation design and development activities, and these can be assisted or hindered by the processes that we use together. What I experienced while trying to design and modify open science instrumentation throughout my PhD was that our actual processes and capabilities leave a lot of room for growth in making such tools easier to customize in practice and to customize in a more deeply collaborative way and to customize as extensively as we want to. So then the research question here is, how might we make it easier to develop open science instrumentation, which would be easier for more people to customize? As a start, we can decompose this big question into two sub-questions. First, how can we make designs which are easier for other people to independently modify and adapt to their own specific needs? For example, to make different design trade-offs between various priorities. And this question focuses on the characteristics of a design snapshotted at a single point in time. And we could start by focusing on the relationship between an instrument's design and the individual person trying to change it. But other difficulties become apparent when we try to extend such changes across larger scales of development. For example, by having more people reuse, extend, and integrate those changes, which have been made simultaneously and independently by various other people scattered around the world. The associated problems are already well recognized in the software industry, as we can see in this quote here. My talk will show how similar challenges need to be addressed when we want people who are independently customizing their own instruments based on the design snapshot from a single point in time to also collaborate and build upon each other's design adaptations over time. These kinds of collective activities, ranging from tightly centralized coordination to something near anarchy, are an important part of how open source projects have achieved a long-lived impact in the software world. But even in software, these changes remain difficult. And open science instrument designs often integrate both hardware and software, which may expose further challenges for distributed innovation and collaboration. So the second question guiding my thesis is, how can we make it easier for manufacturers and operators of open science instruments to extend their own design changes as far as they want, even across large scales of collaboration, design space, geographical space, and over time? These questions were on my mind throughout the open science instrumentation projects that I worked on during my PhD, as I helped to shape their design and implementation. And in those projects, which had started building communities of users, our users wanted to change their instruments in various ways that raised the same underlying questions. So I had to investigate these challenges very directly and concretely through my technical involvements. Due to time constraints today, I'll only cover a subset of my work, and I'll spend more time on some projects than others. To outline the structure of my talk, I'll now give a preview of each project, which I'll discuss in the rest of my talk individually, which will build up to a comparative overview synthesizing the lessons learned from these projects. These projects span a range of application areas, including frugal science tools, medical diagnostic devices, and life-critical medical devices, as well as environmental monitoring devices. And they have different levels of architectural design complexity and hardware-software integration, as well as community size and engagement, geographical distribution, design adaptation, and project maturity. With the first three projects on the lower left, we'll look at some special considerations for modular design as one strategy for facilitating design adaptation. First, the Handyfuge is a hand-powered centrifuge for low-cost disease diagnostics anywhere. Second, the ESPressoscope is a low-cost modular approach for designing digital microscopes, which can be used anywhere and in large numbers. Third, the Squid is a modular toolkit or framework for developing reconfigurable automated microscopes with complex functionalities. Then I'll talk about two projects which pose challenges specific to higher project complexity, including more extended change. The Pufferfish is a reference design for a mechanical ventilator intended to be manufacturable around the world and to be safe enough for long-term use in critical care medicine also around the world. Also, the PlanktoScope is a low-cost flow-through microscope for biological oceanography, and it's intended to be easy and versatile enough for users to rapidly reconfigure to suit their own needs. Finally, I'll talk about a new software tool I developed called Forklift, where I looked at the lessons I learned from the achievements and difficulties I experienced in these previous projects, and then I tried to solve some of the complexity that comes with a class of computerized devices like the PlanktoScope or the Pufferfish, specifically for situations where you want to independently customize and recompose the software embedded in those devices as their device-specific operating systems, and when we want to make those design changes while simultaneously trying to integrate changes being made by other people on an ongoing basis. In other words, software updates. If we hope to realize the vision of open science instruments, which are practical for people around the world to rely on for their work, and with their custom modifications and extensions to those tools, then we need design principles, patterns, processes, and tools which enable better distributed collaboration on design adaptations. And for this, we can learn a lot from processes and tools which have helped to make open source software projects so successful. But we'll work our way towards that gradually over this talk, and instead I'll first start with a hardware project, the Handyfuge. To give some context to this project, there have been many advancements in developing high accuracy assays for diagnosing diseases in a frugal way with low resource requirements. For example, here we see two variants of a LAMP protocol, short for loop-mediated isothermal amplification of nucleic acids, as a test for COVID-19 viruses in saliva samples. Sample processing is a significant part of making such assays achieve the desired performance. One common technique for sample processing is to add tiny beads to samples so that they'll bind to certain components of those samples, and then we separate out those beads by density. For example, the more sensitive LAMP assay here on the bottom includes a step to separate viral RNA from other undesirable components of saliva samples. Typically, this density-based separation is done with a motorized centrifuge to spin the sample tubes very quickly, and conventionally that requires electricity, which limits how frugal a test can be. Many people have wanted to be able to perform such centrifugation anywhere out in the world, even without access to electricity. Along those lines, our lab had previously developed an ultra-low-cost design called the Pufferfish, which you see here, and it consists of two handles joined by a loop of string to spin a paper disc back and forth, like we see here. Then the paper disc would hold two capillary tubes containing samples to be processed. The Pufferfish was really easy to operate out in field settings, and it was able to achieve really impressive centrifugation performance for small samples. But its safety characteristics left room for improvement. Part of the challenge was with this design was that the various components of the overall system were very closely coupled in their interactions with each other. So for example, any safety mechanisms which increased the weight of the spinning paper disc would drastically affect the behavior and centrifugation performance of the Pufferfish. Because we were interested in centrifuging samples with infectious diseases, we wanted an alternative design which could handle larger samples and which could accommodate more comprehensive safety features. So I developed a centrifuge prototype whose design was split into a rotation generation module and a rotor module joined by a spinning axle, as well as an easy way to add safety features through a modular enclosure system. The rotation generation module was made using components taken directly from a hand-powered squeeze flashlight shaped like a bear, as you see here. And as we can see in this video of an earlier prototype, the centrifuge is operated in a very similar way as the flashlights. I was later able to add a rotation measurement module using magnetic sensors in order to measure the rotational speed of the Handyfuge, which allowed me to compare the performance of this early prototype to other designs along these relevant performance characteristics in the design space of hand-powered centrifuges. This performance was practical and useful enough that my lab mate Adam Larson successfully used it for processing saliva samples as input to a LAMP assay for COVID testing. Because we removed the need for a motorized centrifuge this way, now the entire diagnostic test could be done from start to end without relying on electrical infrastructure. What I learned in the course of iterating on prototypes of the Handyfuge was that its modular architecture made it easy for me to try out new ideas for design variants. For example, here we see maybe the first or second version of the design for the rotor module. This was relatively heavy, which prevented the centrifuge from spinning quickly. The modularity of the device architecture made it easy for me to try out different rotor designs so that I could develop a practical real-world design intuition for factors that affect the performance of the centrifuge for making design adaptations. And that led to the version that we used with the COVID LAMP assay. And here's a later experiment with the rotation generation module where I changed that module in order to experiment with the gear ratio of the gear train and the resulting impacts on device performance, which are kind of actually counterintuitive to what I had imagined before building the thing and testing it out. By dividing the system design into two modules, which only interacted through this interface of a spinning axle, so that I could make changes to the design of each module independent of the other module, it became easier for me to generate, test, and iterate on design variants of the overall device. And this allowed me to incrementally improve the system design through a series of small and fast experiments. By incrementally replacing a system's module this way, we can adapt the design to modify its functionality to meet new requirements. And we can do this without having to redesign everything from scratch or just everything together with each change, which would be harder and more complicated. So now that I've discussed how modularity facilitated the process of making design changes in the Handyfuge, I'll move on to the ESPressoscope project, which adds more complexity in the desired variations of device functionalities, and thus a bit more complexity for generating design variants. We were interested in developing microscopes at an intermediate point between the cost, simplicity, and imaging performance of a device like the Foldscope versus the cost, adaptability, and imaging quality of devices like the open flexure microscope from the talk's introduction, or like the Squid microscopy platform, which we'll cover later. At this point in the design space, devices can have interesting digital microscopy capabilities beyond what's feasible with a Foldscope, but such devices would still be affordable enough to deploy in large numbers and to places where they might get lost or damaged, like for several days or even weeks at the bottom of a lake. Given the price and ubiquity of ESP32-based microcontroller boards with image sensors, we thought that such boards could be an interesting core module, which could provide many useful capabilities for many different kinds of affordable microscopes. So we chose the name ESPressoscope for the specific way that we use ESP32 camera boards to design small but powerful optical devices. My collaborator, Benedict Diederich, designed prototypes for 5 different types of optical devices. First, the MatchboxScope is the core 3D printed design for a small, portable, bright field microscope with a Wi-Fi based user interface and a variety of possible module substitutions for more specialized functionalities. Then the Anglerfish is a microscope designed for deployment underwater to do things like time-lapse microscopy, for example, to study the growth of biofilms in aquatic ecosystems on surfaces. This was implemented by combining the ESP32 camera board with modular optical components from an entirely different microscopy framework called OpenUC2, as you can see there with these blocks. The ESPlanktoscope is a matchbox scope extended to add flow imaging capabilities for particles suspended in water samples, kind of like a stripped down version of the PlanktoScope project, which I'll discuss a bit later in my talk. Then the ESPectrophotometer is a digital spectrophotometer for measuring light intensities across visible light wavelengths, and strictly speaking, it's not a microscope. Finally, the Holo ESP is a lensless inline holographic microscope, which enables post-processing to adjust image focus. The designs of these devices may seem independent and very different at first glance, but actually all these diverse designs came from the same architecture of modularity. And it was easy for Benedict to generate these design variants because we had decomposed the overall system into a set of functions, each of which could be implemented by any member of a set of modules, such that the modules could be selected or recombined depending on system requirements. So what we were really doing here was identifying and demonstrating the feasibility of not just a single microscope design or a single microscope framework based on ESP32 microcontrollers, but rather a design pattern for low-cost digital microscopes that could be useful both within our own specific toolkit of modules, but also beyond it, and even with other microscopy frameworks based on entirely different sets of modules and different ways of integrating those modules, like with the OpenUC2 modules in the Anglerfish. So from the ESPressoscope project, we can identify 2 aspects of system modularity as ways of facilitating design adaptation. First, an individual module may be useful and generalizable enough that it continues to be useful in new design variants with very different functionalities, and even with very different technical implementations. Then we can make design adaptation easier by identifying other kinds of modules with similar potential, and then making those modules easy to work with and recombine. For the ESPressoscope, this was the ESP32-based camera module, but we can also see parallels with Arduino microcontrollers and Raspberry Pi single-board computers. Second, the way that we design how modules are to be organized into devices affects what is or isn't made easier in making new design adaptations. For example, the ESPlanktoscope was a relatively straightforward extension of the MatchboxScope design with isolated additions and replacements of modules. By contrast, the ESPectrophotometer's mechanical design is very divergent and basically unique, as the required functionalities went beyond what was supported by the MatchboxScope's system for mechanically composing and recomposing modules. So Benedict had to do a bit more work there in making that design because he couldn't just directly reuse the mechanical designs of modules from the other devices. We'll see that these 2 aspects of design modularity come up again from a different angle with the next project, Squid. Unlike the ESPressoscope, it's a platform meant to facilitate developing modular microscopes with more complex integration of modules to support more advanced research experiments in the lab. At this level of capabilities, usually labs have to buy one or more expensive and highly integrated pieces of equipment which are not readily reconfigurable, like what we see with this structured illumination microscope. Usually these kinds of microscopes consist of optical modules for generating and manipulating and capturing light, electromechanical modules for moving those optical modules and for moving samples, control modules for controlling the optical and electromechanical modules, and some kind of computer which can acquire data and orchestrate everything through the control modules. A flexible platform for control is needed to integrate the various combinations of all the other modules needed for a diverse range of microscope types. So my involvement in the Squid project focused on supporting my lab mate Hongchen Li by designing a way for the control modules to be made adaptable enough to support the envisioned flexibility of the Squid platform. The Squid is now being commercialized as one particular microscope designed by Hongchen's new Cephala company as a spin-off from our lab. But here I'll focus on how we approached the design of the Squid's control electronics during the earlier exploratory phase of instrumentation development, when Hongchen was still generating and testing many variant designs with diverse functionalities to figure out what would make sense. My first iteration of the driver electronics was a printed circuit board with a relatively monolithic design to integrate various commercial boards as submodules. But during my process of laying out the board, Hongchen would naturally think of more things that he wanted me to add to the layout because we were still trying to figure out exactly what we would need. Adding these things forced me to spend a lot of time changing the layout of other parts on the board because of various design constraints that we had that ended up being kind of non-local constraints. Having to redo my work this way was annoying for me. So for the next version of the driver electronics, I switched to a more modular design to reduce the effort needed to make layout changes to match any future changes to requirements for the capabilities and interfaces of the driver electronics. For this, I chose a layered design for integrating multiple circuit boards together by stacking them with connectors so that different functionalities could be implemented independently on different boards. And then the only coordination needed between boards was in the stacking connectors. By choosing this modularity architecture in which we could stack different combinations of boards together, I left open various ways to extend or adapt the driver electronics to use cases that we hadn't anticipated or couldn't anticipate during the initial design. For example, by adding new boards or by replacing existing boards with other versions of those boards. To implement this, I use stacking headers as a uniform connector interface between boards, like what is done with Arduino Shield and Raspberry Pi hats. With this driver electronics system, Hongchen was able to develop a variety of microscope configurations with very different form factors adapted to a wide range of very different use cases of other researchers who wanted to use the Squid microscopes. And they were all using this stacked board system as the core of the driver electronics. And we were able to iterate more easily on the design of the driver electronics during this process. One of these design variants created by Hongchen, called the Octopi, is the application of the Squid modular microscopy concept into medical diagnostics. The Octopi is a microscope intended to be easily reconfigured by operators with different modules as a way of using different imaging modalities for different diseases, such as fluorescence microscopy for malaria or Raman microscopy for tuberculosis. My stacked driver electronics design was the driver platform for a transitional phase of the Octopi's development, bridging between, on the left, the hand-wired boards that we built when I tested two very early prototypes of the Octopi with patient blood samples in Eastern Uganda at the start of my PhD, and on the right, a tightly integrated and more monolithic driver system in the current version of the Octopi, now that Hongchen has tightened and solidified the range of necessary functionalities and suitable components on the path to commercial manufacturing. This driver electronics system also turned out to be useful as a prototyping platform beyond microscopy. So on the left here, we see the same driver system being used to drive some sensors and actuators in a test jig, which Hongchen had built to help with optimizing a lamp-based frugal diagnostic system being developed by our lab. And on the upper right, we can see Hongchen using this same driver electronic system to build a prototype for a critical care ventilator during the first year of the COVID pandemic, which I'll discuss further in the next section of my talk. Finally, on the lower right, we can see the same driver electronic system being used with a different configuration of board stacking in order to test some pumps and airflow sensors for that ventilator project. This reconfiguration was only possible because of the layered board design I had chosen to enable modular extension. By integrating the driver electronics in a way that bridged the design space between, on the one hand, the really generic reusability of a microcontroller board like the Arduino, and on the other hand, the very application-specific specialization of a monolithically integrated circuit board, the result was a system which was reusable and useful as a self-contained module in its own right for prototyping and evaluating new design variants of microscopes and even of other kinds of devices too. Even though my driver system design was specialized for microscopes, it was also able to remain somewhat generic because it was open to being arbitrarily changed by reconfiguring the boards layered into it through a uniform interface of stacking connectors, which reduced the effort needed to add new functionalities later. So far, we've been looking at processes of design adaptation where one person in the present tries to collaborate with a future version of themselves, or where a small number of people are working closely together to collaborate with each other in early prototyping. Now I'll move on to Pufferfish, which is a project with a significantly higher level of complexity, both in its ambitions for collaboration and in its technical design as a critical care ventilator intended for long-term use in clinical settings. The COVID-19 pandemic has exposed many barriers to producing critical medical supplies and equipment in a way that would guarantee equitable access to those products around the world. People have started efforts to try to reduce these barriers, such as by developing open source ventilators for local manufacturing around the world. But the devices that they were able to design on such a short notice, like the AmbuBag squeezer we see here on the left, weren't really safe enough for actual clinical use. On the other hand, a previous project, the open artificial pancreas system for people living with type 1 diabetes, had already demonstrated safe usage of an open source medical device produced by distributed manufacturing. So we wanted to develop an open source reference design meeting FDA standards for a safe critical care ventilator, which would also be appropriate for clinical use beyond the COVID pandemic and beyond the US. The idea was that manufacturers would adapt this design according to the context of their local markets, such as their local supply chains and their local clinical needs. So our approach was not to just make a new device design and then send it to manufacturers through knowledge transfer, but instead we built co-design partnerships with people around the world, including some mentors from companies with extensive experience in safety critical systems, including medical devices and self-driving cars, as well as manufacturers in Kenya, Nepal, and India who wanted to locally produce ventilators but were new to medical devices. Bart Forge was deeply involved in developing the reference design's enclosure and mechanical structure, and they successfully replicated our prototype reference design in small-scale production, like you see in that video. The NIC in Nepal was part of the team that designed the electrical systems, and NJ8 Technologies made their own adaptations beyond the reference design, for example, adding a larger external battery to account for their local electrical infrastructural conditions. Because our Pufferfish ventilator's overall design required the full level of functionality and safety which clinicians expect from critical care ventilators, our project's technical design also became relatively complex. For example, the pneumatic systems, which you see on the right, they had to implement appropriate airflow control and sensing for a variety of ventilation modes and safety risk conditions, as well as mitigations for certain potential failure modes of the system. These requirements in turn generated many requirements for the software that would control our pneumatic sensors and actuators. Similarly, our electrical systems also had to manage safety risks. For example, because our device was in control of every single patient breath, we had to properly mitigate loss of external power. These kinds of requirements created additional requirements for the software. Finally, safe operation of the ventilator required a user interface which would help really busy and multitasking clinicians to understand patient conditions and then make appropriate decisions quickly. And the clinicians would also need to have enough information to take corrective actions appropriately in case of a technical problem with the device, for example, a software failure. Other features also had to be implemented, including various safety check procedures and things like battery lifespan monitoring. Because of all these decisions in hardware and UI design to rely heavily on software for implementing various safety mechanisms and safety checks, a lot of complexity was pushed into the software. I ended up with decision-making and team lead responsibilities for the software architecture, design, and implementation, so I was right in the middle of this complexity. And beyond the safety requirements being generated by other teams for us, we also had our own system requirements for safety that we were identifying. For example, we split the software into separate processes running in different domains with different levels of complexity and different failure risks and different amounts of safety critical functionality in order to limit the blast radius of certain kinds of software failure from affecting really life-critical functionalities. But this meant that I also had to design a communication protocol between these modules with appropriate safety characteristics, such as in the reliability and latency of information transmission. And the hardware and the user interface and the list of required functionalities were all simultaneously evolving throughout the project. So the requirements for software started to change in ways that were challenging for some of the key decisions I had previously made in the architecture. For example, my initial design for trying to keep the communication protocol simple. Because of the continually increasing complexity of software requirements, our project reached indefinite hiatus before we were able to complete a prototype implementation of the necessary software systems. The lesson here is that for devices as complex as ventilators, we really need a sustained process of laying technical foundations as working systems, which could be reusable, adaptable, and composable, and which could hopefully simplify future device development efforts in the next crisis. This is first and foremost a challenge with organizing people and processes upstream of challenges for creating new technical artifacts. We did have successes with people and processes in this project. We helped companies from the military weapons and automotive parts industries to start learning how to manufacture medical devices for their local markets. Bart Forge initially joined with an expectation of a more unidirectional tech transfer process, but as we got them involved in developing the reference designs hardware, they were able to take increasing responsibility. And our manufacturing partners were able to independently make their own design extensions and adaptations, and to work together from across disparate time zones and markets in collaboration with the academic labs that were involved in the project. So we achieved a proof of concept of distributed design and manufacturing of a medical device by commercial entities collaborating to develop a shared open source design. We can also learn from the challenges we faced. As requirements for the overall device became more and more complex, software complexity increased much more than hardware complexity. Effective capacity building for open science instrumentation may require closer communication and co-design between people working on the hardware and people working on the software, especially when the hardware design and overall system requirements are constantly changing in ways that have serious consequences for a software architecture. Continuing on this question of people and processes, now I'll talk about the PlanktoScope project to look more closely at the extra collaboration complexities which appear as a project tries to scale to a broader community of users who are all trying to do very different things based on the same device. As a low-cost microscope for measuring plankton biodiversity, the PlanktoScope has been receiving strong interest from many scientists and local communities. This is because of the key importance of plankton for our planet's ecosystems, not only because they're collectively responsible for roughly half of the world's total absorption of atmospheric carbon dioxide and processing to produce oxygen, but also because they're the base of marine food webs, so that they're the ecological foundation of communities who rely on the oceans as a source of food. Plankton populations are incredibly complex and they vary a lot over space and time, but we only have a really limited grasp on their dynamics. For example, the bottom half of the slide shows global coverage of the Argo Float program for monitoring a bunch of physical parameters about the world's oceans, like temperature, salinity, conductivity, with a large network of floating sensors. By contrast, the upper half of the slide shows the very regional coverage of the Continuous Plankton Recorder program for monitoring plankton biodiversity. They do this by towing devices behind commercial ships, which capture plankton on a silk mesh, so that someone can later sit in a lab all day and identify and count the different types of plankton caught on the mesh. This is a very labor-intensive process, so more recent microscopes have been developed to automatically save images of plankton in water samples as they flow through a tube, so that those images can be analyzed later through a semi-automated software process to make quantitative measurements about plankton populations. But as you can see in the middle here, these instruments are all very expensive, which prevents them from being deployed at the global scale that we need for monitoring plankton biodiversity. The reason that so many people have been so excited about the PlanktoScope in particular is that it provides this capability at a much more affordable cost, ranging between a few $100s if you're really DIY to a few $1,000s if you want the nice version. This enables the possibility of a larger scale microscope deployments around the world. And because of the PlanktoScope's open source design, people around the world have been building their own units. And we've also been seeing people making various kinds of hardware design modifications to suit their own requirements. As the community continued growing, it revealed some pain points around the PlanktoScope's software, which runs on its embedded computer. I experienced some of these difficulties firsthand when I was part of a research expedition in the Arctic Ocean, northwest of Alaska, to study where all the surface plankton here were going with the carbon dioxide that they had absorbed from the atmosphere. And we operated a PlanktoScope intensely on a very large number of water samples being collected at any hour of the day, almost continuously for 45 days. For example, because the device wasn't originally intended to be used for handling so many samples back to back, I had to improvise some additional software scripts and operating system configuration changes for a very non-standard networking setup involving a second computer so that I could have a system to transfer, manage, and process all the data while the PlanktoScope kept acquiring more data. My customizations here would be pretty useful for other people too, but because of how the PlanktoScope's operating system was built, there wasn't a straightforward way for me to share my customizations for other people to reuse directly with their own PlanktoScopes. But this hack was needed so that we could collect as much data in the Arctic as was planned for the expedition, which was an amount that significantly increased the number of samples imaged with PlanktoScopes in the online database of plankton images, which the PlanktoScope is designed to integrate with. But we wanted to increase our data acquisition throughput even more by increasing the deployment scale of PlanktoScopes so that we could eventually have a network of instruments all continuously observing plankton populations in the ecosystems where they were deployed and continuously making that data available online for anyone to use, in other words, open science. This would require further extensions to the PlanktoScope's software capabilities. So as a first prototype towards that goal, we built a rack of 8 PlanktoScopes, each attached to its own aquarium as an experimental ecosystem, and then we connected each one to the internet. In order to enable these PlanktoScopes to be accessed and operated remotely, but without having to do all the work of rewriting the PlanktoScope software to support this very different network architecture than what the software was originally designed for, I deployed a mesh virtual private network, which allowed other computers on the internet in the same virtual network to connect directly to the PlanktoScopes as if they had all been connected to the same Wi-Fi hotspot or router. This way, I was able to access the network APIs and web browser user interfaces, which normally were only accessed over direct physical, whether it's Ethernet or Wi-Fi connections, to the PlanktoScopes. I also added a remote file system so that the PlanktoScope software would upload its acquired images directly to cloud storage, but without me having to do the work of rewriting the relevant part of the PlanktoScope software that saved data. These changes required me to modify the operating system running on each PlanktoScope to add additional programs, to change some operating system configuration files, and to fix some bugs in the PlanktoScope software itself. Although I got to a working prototype of the entire system, my prototyping progress was hindered by various aspects of the PlanktoScope project's software and processes around software. These things were experienced as issues in the project only after I began trying to make software changes to this new extent that previously hadn't been accounted or designed for in the PlanktoScope project's software and software maintenance processes. These issues occurred because the PlanktoScope project's modularity and ease of configuration had been primarily focused on its hardware. It was only in the course of trying to extend the PlanktoScope software far beyond its original intended design and using the PlanktoScope very intensely that I could identify these specific implementation challenges for the project. What I'm focusing on here is about the goal of growing a community of people who would make design adaptations and extensions in the PlanktoScope software, then share those changes with others, and then continually reuse and integrate each other's improvements. The specific technical difficulty with this was that we needed an easier way for users to combine different kinds of software changes from different kinds of sources, for example, from the overall project or from other users, on machines which are already accumulating other design changes made by the PlanktoScope's own operator. This problem has not yet been satisfactorily solved in real-world software systems. As we can see in this photo I took of a sticker on a piece of really expensive-looking scientific equipment, where changes from Microsoft were suspected, probably with good reason, to carry a risk of disrupting whatever non-Microsoft software was installed on that machine. So I tried to solve this problem for the PlanktoScope's operating system by synthesizing what I had learned from my previous projects about modular decomposition and interfaces, layered integration of modules, and other ways of exposing design patterns to organize modularity, the role of reconfigurability in making things generically reusable, challenges with software complexity, and finally, the need to improve coordination on software development with the mental models that hardware developers have for the devices that we're working on. The result is a software tool called Forklift, which is a working prototype of a broader approach to software composition, software extension, software configuration management, and software updates through a bills of materials abstraction. So the common hardware paradigm which Forklift extends and applies to software is the bill of materials, or BOM for short, which normally is just a list of all the hardware parts needed to build a device. But if we specify our BOM as a list of modules with enough additional information about each module's interfaces and any other constraints for how modules can be combined together through those interfaces, then that information could, in theory, fully specify a unique configuration for combining the modules together into a working device. From the BOM, we could even maybe computationally generate the assembly instructions needed for building the device. And if we were really good at robotics, better than anyone is right now, then we could even make a robot that would execute any assembly instructions that were generated. After we adjust the design and snapshot it as a new version, for example, V2 on the right, then our changes are represented as a new BOM. We can also run automatic design rule checks to test whether there's actually a valid way for the modules to connect to each other according to simple rules and constraints associated with those interfaces. For example, if our BOM had a full-size HDMI cable and we had a computer which only has a micro HDMI port, then we could computationally determine that the design can actually be built without adding some kind of a port adapter for HDMI to the BOM. We can easily compare any two BOMs to see what modules are in both designs and what modules must be added or removed to go from one design to the other in one direction or the other. From that comparison, we can then compute a plan for how a physical device matching one particular design would need to be modified in order to make it match a different design. And if we had a really fancy robot, then we could even make it execute that plan. Forklift translates this idea from hardware into software so that we can specify a device's operating system as a set of modules with particular interfaces and constraints on how modules can be combined through those interfaces. Then starting from a computer with an operating system which matches one BOM, Forklift can automatically modify it to match any other BOM. So far in Forklift, I've implemented two types of components for operating system modules. First, a software program can be specified as one or more Linux containers, which are self-contained and isolated bundles of programs, along with everything those programs need to work. As an implementation detail, I'm using Docker right now, but that doesn't necessarily have to be the case. Second, an executable binary, a script, A system service definition, a configuration file, or really any other system file can be included in the BOM or specified in the BOM as a download from the internet, so that Forklift will make it available for integration into the operating system at a specific file path. If the base operating system already had a different version of that file at that path, then that other version would be ignored as long as the Forklift provided file exists. On the other hand, if the user tries to edit a file at that path, then the operating system will automatically make a copy and save that new version to a different place, which will cause the Forklift managed version of the file to be ignored. In this way, a user can make their own customizations, which override what's provided through Forklift. With this file overlay mechanism provided by Linux to separate out all these different kinds of changes, so that different versions of files are all stored in the file system, even when only one version of that file is actively being used, we can easily and safely switch back and forth between different versions of files, for example, through operating system upgrade and downgrade processes. In Forklift, our BOMs are called pallets, so that we have a name that you can safely say at the airport. And pallets are version controlled and distributed online as Git repositories. So this is the model that's familiar for software people. In the current stable version of the PlanktoScope's operating system, the parts which would be useful for non-PlanktoScope devices are all composed and specified using Forklift. As a result, an independent project, the OpenUC2 modular microscopy framework on the right, which is developed by my collaborator Benedict from the ESPressoscope section of this talk, that project now relies on Forklift to directly reuse a bunch of BOM modules which I had developed specifically for the PlanktoScope. This saved him the work of having to figure out and re-implement those same functionalities himself. So he could get from his idea to like something just as functional as a PlanktoScope much more quickly. This was accomplished in OpenUC2 by installing the Forklift tool and integrating into the operating system build process, and then creating a few configuration files in a new OpenUC2 palette, in other words, a new Git repository, to import certain groups of module declaration files from the PlanktoScope palette. The OpenUC2 palette also overrides some of these imported files through a separate customization layer of files stored in the palette, but that's kind of way too in the weeds for this talk. This way, the OpenUC2 project was able to integrate improvements that I was making in the PlanktoScope's operating system, even as OpenUC2 was making their own changes, including changes to their overrides for some of the same files in modules that I was simultaneously changing in the PlanktoScope project. In this demo, we can see that all we need to do to change a running Raspberry Pi computer, which is being recorded here, from one palette to another is to run a single command and then reboots. Here, we're switching the Raspberry Pi from a minimal demo with just kind of like hello world webpage into the PlanktoScope's operating system, and we could also switch back just as easily. This way, Forklift makes it much simpler to upgrade or downgrade the operating system, or to safely try out some changes or customizations made by us or by someone else. If we then try to add or remove a module to the palette, which would cause a conflict with another module or a missing dependency with the interface of another module in the palette, then Forklift will run its automatic design rule checks and prevent us from applying that change by giving an error. This way, Forklift enables us to try out new combinations of changes from different people, maybe people who don't exactly know what they're doing, but we can try it out with much more confidence because a certain class of common bugs can be entirely prevented by this system of constraints between modules through their interfaces. I'm still iterating on Forklift's design to make it simpler to explain and more mature for broader use, but now it's already running as part of PlanktoScopes and OpenUC2 microscopes, which people are using. And in a few weeks, I'll be starting gainful employment with OpenUC2 to continue this work on Forklift and on OpenUC2's operating system as part of their goal for making digital microscopes and other optical devices easy to adapt through reconfiguration and recomposition in both their hardware and the software together, which is kind of the spirit of this talk. More broadly, wherever we have integrated hardware and software systems which need to be reconfigurable and adaptable in processes of extended change over time between people for whom tight coordination would be difficult, I think this recomposable BOM pattern, which Forklift demonstrates, could be useful in helping both hardware people and software people to collaboratively experiment with design adaptation more easily and with more confidence. My design of Forklift is really the synthesis of everything I've learned in trying to develop open science instrumentation through this PhD. So now I'll return to the motivating question I had presented at the start of this talk, which is about how to enable processes of design change, which can be extended across large scales of time, space, and collaboration in developing open science instruments, which are meant to be adapted by people who need them for new or different purposes. We've seen today that the technical design of the system's modularity can strongly shape what processes are available for people to try to collaborate on evolving that design towards new directions. We've also seen that processes, tools, and patterns are an important area for technical improvement to address the challenges that we face with realizing the potential of open science instruments. And finally, in the context of open science instruments, which combine hardware and software, we need to pay attention to how people work in each domain so that we can help them design instruments where the hardware and software are easy to evolve together. I was only able to design a system like Forklift to solve process-related problems for an entire class of instruments because of the perspective that I gained from being deeply involved in the technical development of a wide range of projects across both hardware and software and across a variety of other technologies and application areas. By taking a similar comparative approach in other projects, I think future researchers will also be able to identify all kinds of other insights from their projects for improving the technical aspects of how we as people can get better at working together to develop the kinds of open science instruments that will hopefully transform how open science is done towards a more broadly participatory and equitable path. My work is currently being relied on people running some of these open science instruments for their own work, including their own research. And in my future work at OpenUC2, I'll be continuing to investigate and address the challenges which I've discussed throughout my talk today as we try to grow that community of people using and innovating with OpenUC2's modular optics framework. So the work I've shared today was only possible due to research support and help from so many people around me. So this is especially the Prakash Lab and so many people here who are not in this photo. Manu, Jasmine, and Shama, who really made it possible for me to do my work in so many ways. Hongchun, Deepak, and George, who were kind of my mentors for many of these projects and also collaborators for some of them. As well as Adam, Anessa, and Pranav, who are collaborators on various projects. And Olivia, Jonghyun, Abby, Shreya, and Ching, who helped me out with my tenors project when it was extremely difficult for me to keep making progress. Finally, there's Grace, Melanie, and Hope, who provided me much encouragement throughout parts of the PhD where I needed the encouragement to keep pushing through on the PlanktoScope and the tenors project. And then Charlotte, Hazel, and Bennett for doing so much to build a lab culture. And then all of this work was only possible because of extensive collaboration with so many people around the world. So in the PlanktoScope project, there is the Fairoscope company, which spun out of our lab to commercialize the PlanktoScopes. There are these Fabian and Satoshi, who were really involved as people in the community, sitting in on meetings where I was thinking through some of these design changes for the PlanktoScope and providing extremely crucial feedback and testing for my work. And then there's also this other project that I didn't mention for trying to use PlanktoScopes to monitor local ecosystems as an early warning system for fish poisoning, which would affect the local community in this part of Indonesia. And so people there really helped me with having a good learning experience as a person training people in this workshop. Then the Arctic Plankton project was only possible because of all these people here. And then Benedict was my co-conspirator on ESPressoscope and Forklift, and now in the few weeks on OpenUC2. Pufferfish, we had so many mentors and people who helped out with the software. And then with the Octopi, there were these people in Uganda and from the US working in Uganda who really made it possible for me to do my work in testing the Octopi. And then finally, we had a collaborator in exploring another application of Handyfuge, which I did not mention today. Finally, this work was possible because of the funding that was provided to me throughout the PhD. And of course, thanks for the Defense Committee so that I could be up here today talking to you. So with that, now I'd love to hear any questions or comments that you might have.
Maybe we can do the open round of questions, but I know many of you use but I can talk about it. Well,
yeah, this is really exciting to see all these different types of projects, and I'm wondering, one dimension of collaboration is the level of experience of the collaborators. So I wonder if you can talk just a little bit about that and about how you're thinking about that with respect to Forklift.
Yeah, I think the place where I had first-hand experience with this was in PlanktoScope, where some of the software contractors that Bart Forge had hired to help me out with implementing the software were like,
You mean Pufferfish?
with Pufferfish. They were not very experienced in the kind of software development that we were trying to do. And that we didn't really figure out how to solve that, which was maybe part of the problem. I think part of what I was trying to address in designing Forklift is providing different levels at which people could try to work on making their own changes and adaptations, so that simpler changes could be done in a more simple way that would be more broadly accessible, while more complicated changes, which required maybe deeper mental models of the software, could still be done, but maybe they were harder to do.
But you could not put together an incompatible configuration, which was a big risk for us.
Yeah. So I think a lot of pitfalls that people often run into in software could be solved with tools that kind of provide guardrails for them. So that would be another aspect of this.
Thank you.
Other comments, questions?
Hi, amazing work. Ethan. Like one question that I'm wondering how your thoughts on this. So a lot of open source software just doesn't get deployed. I feel like its users don't know how to use it themselves, don't know how to implement it. So there's requirement for that kind of user base or user community to guide that. Do you have any thoughts on this? How you can do that for people innovation?
Yeah, so I think there's low hanging fruit, and then there's things that I'm not super confident that I would be able to answer. I think low hanging fruit is like stuff that I've not yet done with Forklift, like actually documenting the whole system and providing guides and tutorials. I think one common theme I've seen with software projects is that really the person who's maintaining the project kind of also needs to be a really good customer service person in expanding like the number of people who are initially trying things out, providing all the support that they need to succeed with their own projects. And then it's about kind of building a community that will help each other out as the community grows.
From PlanktoScope users online. Last chance for a question, and then we're going to transition to your story. Okay, I think what we'll do is maybe let's do this more personal acknowledgements and other sites, and then we'll together.
Yeah. Yeah, so I'm only able to be here today sharing this work because of the support and efforts of so many people who've made my life possible. Without them, I wouldn't have made it this far. So if I try to give a full accounting, then we'd be here forever. But that means I can only give a very incomplete list of acknowledgements. So if I didn't include a photo with you here, please know that I'm still very grateful to you, including if you are watching a video recording later. Oh. Okay, so these are my parents. They definitely may be the kind of person who would try to do a PhD. And so they're a key part of all of this work. I guess the images are going to take a while to load. This was my nanny from the few years between when I was born in the US and when I moved back to the US in Beijing. So she was an important part of my emotional development as a person. There were so many people in my life before the PhD and various kinds of communities. I haven't done the best at keeping in touch with them, but Mark on the lower right here, this was a photo of us from 2 weeks ago when I was up in SF to finish my German visa application. So it was nice catching up, and I hope to cross paths with other people from all these other communities in the future. Next, we see here on the right corner, Ross, and on the left corner, Pavna. They were really crucial in my development as a person who would be interested in the kinds of things I've been talking about here today. And I owe so much to both of you for everything starting with like junior year of undergrad. Next here is my former housemate, Paul. who we went through COVID together. So he tolerated all of my late night calls with people from India working on the Pufferfish project. And for that, I'm very thankful. Next here on the left is Melanie, who was my hiking buddy while she was part of the lab. So we went on many fun trips together, and we also organized camping trips with other people from the lab. So here we see Hope and I am blanking on names, sorry. Pranav holding the camera, not visible here. Yeah, so this was a really meaningful part of my experience in the lab. Then here we have Grace. This was a photo from like 1 or 2 weeks before we started the BioE PhD program. And now we are kind of helping each other cross the finish line here with our defenses. So thanks so much for the encouragement and support. And then more recently, there are a bunch of people at the Mountain View Buddhist Temple who kind of welcomed me into their community, and that really helped me make it through this year and gave me a sense of place and belonging. And finally, this is a park about like a half-day hike south from campus. These are lands of the San Francisco Creek watershed and the unceded ancestral homeland of the Puchon and Opon tribes of the Ramaytush Ohlone people. And so they're identified today by the Association of Ramaytush Ohlone and the Mwekma Ohlone tribes as the lands where they have ancestral ties. And this land was really like a key part of how I was able to kind of have sanity during the past half of my PhD. And this is also where we do our research. So my talk today is really a way of expressing gratitude to everyone working to take care of life and land, and I hope my efforts can eventually help to make their work easier. And finally, thanks to all of you today for being here with me. Yeah.
Okay, we're going to transition to the closed part of the events, and it's in time, it's going to be tight. And if you want to just take a 5-minute break, that's good, but we'll start right on. So Julio has picked up my